I want to know, have you studied structure and function of ribosome in other living? In the initial slides you showed the ribosome in other living things, plant kingdom and animal kingdom. You showed one slide.
Yes.
I want to know how far you make comparative study of ribosomes. Have you studied comparatively ribosomes in structure and function in other living beings and other human living beings?
Yes.
Genetically, I want to know whether it is evolving.
Yes.
Is some
That's a very
entity
good
which
question.
is evolving gradually in the human form.
Yeah. That's a very good question. So, with crystallography, it was very difficult to study ribosomes from other organisms because it takes many years to get a crystal, and we were actually lucky to get it. But now there's a technique called cryo-electron microscopy, which allows you to get atomic structures even without crystals. And using that method, people have gotten ribosome structures from a variety of species. For example, from yeast, from humans, also from plants, from mitochondria, which are organelles in our cell, which are descendants of bacteria that were absorbed by our cell 2 billion years ago. And these mitochondria have their own ribosomes, and they're no longer like bacterial ribosomes, even though they're descended from bacteria. They've evolved to be completely different from bacterial ribosomes. So we're getting a very good idea of how ribosomes have evolved through the ages.
Some of the scientists in America felt that they were thinking about the ribosomes for comparing to the Vietnam. Now the success of ribosome was proven. Did they change their opinion about the ribosomes after your papers?
No, sorry, I didn't understand the first part.
Okay. In the early part of this invention, with regard to the ribosome, some of them felt that they were granted Vietnam War. Actually, when Vietnam War was going there, what they felt it was mentioned by you in the book.
Yes.
Now ribosome was proven, the significance of ribosome proven for you.
Yeah.
Okay, did they feel the significance of the ribosome?
No, I don't think, the biological science community, the Vietnam War was a political thing going on in the background. I don't think it had much to do with this. Holistic practices for crops. But there's no reason why, for example, governments like the Indian government, through India has many agricultural research labs, why they could not generate their own varieties for use, for example, in India. And the reason that I think it's a good idea is because you can modify crops so that they're more drought tolerant, they're more pest tolerant, so that you don't have to use chemical pesticides or chemical fertilizers. And you can alter the nutritional value of the crop so that you can make, for example, rice or millet or wheat more nutritious, perhaps higher in protein, or have different composition. So there are many possibilities for genetically modified crops that can be actually very beneficial. And the human population is still predicted to increase up, to go up to maybe 10 or 12 billion before it stabilizes. And with climate change and drought and things like that, we really do need to address the food security problem. So I think scientists can play an important role, and it doesn't have to be left to a few multinational companies to do that.
Hello. First of all, at the outset, I would like to thank you, sir, for giving such a wonderful, nice, and lucid presentation of the Nobel. We people sitting in this auditorium are really gifted to look into see the lecture and the struggle that you have undergone over the years, right from your childhood up to today's level. So, my heartiest congratulations to you, sir. And I am also very pleased to talk to you or interact with you And I have one question, sir. Sir, the separation of the ribosomes or in identification of proteins in the ribosomes, we normally go in for X-ray diffraction techniques, right, where you identify different spots, amalgamate them, and then get into different structures. But my question is very simple. Why cannot we go in for the protein purification techniques, protein analytical techniques, like the HPLC analysis or UPHLC analysis or LCMS, like that?
Yeah.
So that we can get better picture about the types of proteins that are present in either 50 years or in 30 years, like that. And also we can assemble them together, delineate what are the different types of proteins present in a given situation.
Yeah, so actually that was all done before the X-ray structure. So when we had the X-ray structure, people already had purified all the proteins using chromatography, just like you say. They had sequenced the proteins so they even knew what the order of amino acids is. The simplest explanation is, if you want to know how a ribosome works, you need to understand how these proteins are located in 3 dimensions, and also the ribosome is 2/3 RNA, so you need to understand how that RNA is folded in 3 dimensions. What you're saying is, let me give you the car analogy. You know, if you went and simply took a car apart, let's say nobody knew what a car looked like, okay, but somebody came and smashed a car and you got all the parts, but you didn't know how the parts were put together, then you would have no idea how a car worked. You know, you would know what a gas pipe looked like or what a steering wheel looked like, but unless you know how it fits together, all, you know, how they're all connected inside a car, you won't know how it works, and that's the reason for this.
Yeah, another question, sir, please. How the drug actions, you say that you have got a lot of antibiotics, and how do you feel that these antibiotics would be interacting with the ribosome, and in what way it is helpful in going for a new drug development?
Yeah, so the reason, when you know, so it was known for 50 years that these antibiotics attack the ribosome, that was known. But what was not known is how they do it. So with the structure, we know exactly where the antibiotic binds on the ribosome. And by knowing that, and knowing the chemical interactions it makes, you can also figure out what function of the ribosome does the antibiotic stop, okay? So you learn a lot about ribosome function and antibiotic action. But then knowing those pockets, you can try and design other molecules that will fit that pocket even better than the natural antibiotic. And that might help overcome resistance, you know, to antibiotics.
Is it possible to modify the structure of ribosome by using drugs?
Very good answer.
Let him answer it again. Sorry?
So is it possible to modify the structure of ribosome by drug-drug interaction or the drug-to-protein interaction?
It's much harder, you know, if you modify the structure of the ribosome, you'll almost invariably make it worse because it has evolved over millions of years. Rather, what you could do is you could modify the antibiotic molecule so it fits the ribosome better.
Venki, when you left India in 1971,
Yes.
you would have had some idea of the state of science, at least in those fields that interested you. I mean, if you can look back over that period today, how far have we come in terms of advancing in science? I know it's a large question, but whatever insights you can provide and what needs to be done.
So I have to tell you, when I left, I was only 19, and I had no real connection with Indian science or Indian scientists. So, and I visited India only a few times in the next 30 years when I lived in the US, only about 3 times in 30 years, and mostly to visit my parents. So I don't know much about what went on between 1971 and 2002. But 2002, my first visit to India was to Madras, when I was asked to give, G.N. Ramachandran had died the year before, and I was asked to give the first G.N. Ramachandran Memorial Lecture, which was held at the Gindi campus of the University of Madras. And that's when I first met Indian scientists. And so I was favorably impressed by some of them. And on that visit, I also went to the Indian Institute of Science in Bangalore for a day. And I was quite, favorably impressed by what I saw there. I'm often asked this question, and I would say that India had a period before independence when it produced world-class scientists. For example, C.V. Raman, J.C. Bose, S.N. Bose, Meghnad Saha, Homi Bhabha, all of these people, and Chandrasekhar, but Chandrasekhar did most of his work abroad. But I'm talking people who actually worked in India and did world-class science, okay? So there was certainly a tradition of excellence. Now, after independence, it persisted for a while because Nehru was very interested in science, and so he funded, helped fund a lot of science institutions and so on. But after a while, I got the feeling that it sort of stagnated for a while. Then, I think, thanks to people like C.N.R. Rao and others, who argued for more science funding and, better facilities, better equipment, things started to improve again. And, there was one thing that happened during this time was science also wilted in the famous state universities. For example, Madras University or Bombay University and so on, or even my own university, Baroda University. And science research rather migrated to central universities and central institutions, like the Indian Institute of Science or the big central institutes. And so that, I think, meant that people who were being educated, like undergraduates and PhD students, were somewhat isolated from where the big, main research was done, and that also wasn't good. And I think, thanks to C.N.R. Rao, there was an attempt to correct it by forming the ICERs, which are undergraduate and postgraduate institutions, which combined education and research in the same institution. So that's another optimistic look. But ultimately, there are a number of factors which holds back Indian science. One is funding. Funding is, Indian science funding is much lower in terms of fraction of GDP compared to other sort of advanced science countries, and much lower than China, which has made tremendous investment in science. And it's actually showing now, because now you can open up journals and see world-class science published by Chinese scientists. And you don't see that so much from India. You have pockets of excellence, but not widespread. And another is bureaucracy. It takes a long time to get anything approved. Then you have all sorts of restrictions on how the money can be used, and even when the money arrives, it takes a long time to even spend it. Then sometimes money is given for big fancy equipment, but there's no money to maintain it. So these are all drawbacks in the system. And so I think Indian scientists are working, I would say, under a handicap. And if it needs a real commitment from government and from society. Society has to understand why science is important for prosperity. I'm going to talk about that tomorrow in Delhi, actually. Because there's a direct connection between knowledge-based societies and prosperity. So countries like Switzerland or Singapore have almost no raw materials or natural resources, but they're extremely wealthy because they've invested in knowledge and technology. Other countries, for example, African countries or even Russia, are extremely rich in natural resources, but they're not that prosperous economically, because they're not as advanced, scientifically And so I think there is a real connection, and governments need to have a vision for 10 or 20 years and not about the next election.
Thank you for that talk. It was both lucid and very inspiring. I have a question about the recent developments in the field of artificial intelligence. You know that AlphaGo has and other packages like that have revolutionized. I don't know. I would like to know what you think is the impact of artificial intelligence on the field.
Yeah, I think artificial intelligence is going to have a huge field, even in my field of protein structure, molecular structure. AlphaFold is a program using artificial intelligence that, given that sequence of amino acids, can predict what the three-dimensional structure would look like. And it's made tremendous advance, and lots of biologists are already using it in their studies. Similarly, you know, a lot of things that we now do painstakingly, you know, by writing programs or even visually, can be done routine, you know, faster and possibly better by artificial intelligence. For example, in genetics, if you, let's say, you know, you have a population, you know, which has cancer, you know, you can, for example, sequence, you know, thousands of millions of people, and then figure out, you know, which among them have which diseases or propensities and so on. And that could be very, very complicated, but you could, with artificial intelligence, perhaps, you know, tease out what are the important factors that, you know, confer certain traits. So I can see lots of huge uses for artificial intelligence, but it has to be done in a careful way and not sort of taken blindly.
What is your reaction to the field? I mean, to the development of AI tools? Will they displace people? What do you think about that aspect?
Sorry, my reaction to what?
To the fact that they might be displacing people from.
I think, you know, it's probably true of, you know, that has been true of every technology right from the Industrial Revolution. In fact, what people don't realize is for the first 100 years of the Industrial Revolution, common people were much worse off. Okay, they had been thrown out of their jobs, all these weavers and, you know, manual workers. They had been thrown out of jobs because of industrialization. And it took 100 years before the prosperity actually improved overall. Okay? And so every disruptive technology will initially make people possibly worse off. But then what will happen is you'll create new industries and new jobs. So humans will probably end up doing new types of things. You know, so we won't be doing, you know, if something can be done very routinely by a computer, then we won't be doing it. We'll be doing something else. You know, but what that is, I don't know. But the transition can be quite difficult. I accept that.
Last question. Sir, I asked you sir. இன்னைக்கு வந்து இருக்கக்கூடிய இளைஞர்கள் வந்து அறிவியலையும் நோபல் பரிசு இதை நோக்கி வந்து இன்றைய இந்திய இளைஞர்கள் வந்து எப்படி பயணிக்கணும்னு நீங்கள் கொஞ்சம் அறிவுறுத்துறீங்க. Like that, what is your suggestion that today's Indian youngsters, how should they move forward towards science and Nobel Prize? What is your advice?
Yes. So the first advice I would say is don't aim for a Nobel Prize. Because the Nobel Prize is, as I tell you, it's a bit like winning a lottery. You know, you would not advise a young person to say you should grow up and win the lottery. Okay, and so that wouldn't be such good advice. But I think what you should do is you should think about what interests you and try to get the best training for that interest. Okay, so if you want to go into science, try and acquire a broad background, which means physics, chemistry, biology, and mathematics, you know, which are the core foundations of all sciences. So you should try to get a good background before you specialize. in whatever interests you. And try to get into the best college you can or university you can, because you'll be surrounded by brighter people, your professors might be more knowledgeable and so on. So at each stage you have to just try and give yourself the best shot. Okay, there's no magic recipe other than that.
You reacted to the Indian Science Congress in 2016. It is a circus.
I suppose I'll never let that down.
Hello? How do you see the promotion of pseudoscience in Indian Science Congress?
Well, I don't want to get too hung up on the Indian Science Congress, and I'll tell you why. Indian scientists that I know, for the most part,